would go into generation of product from microorganisms, or using microorganisms to generate pharmaceutical products. Okay, because remember, first you need to have the microorganisms. If you're from your biotechnology, you know that some of these microorganisms may have to be modified, okay, to facilitate production of these products, okay, or even for like the one we are using, some of them may actually be modified so that as much as they are still producing the same product, the quantities that are being produced are produced actually in higher quantities, okay. So just as an example, we will look at benzyl penicillin, but of course you're aware that microorganisms will be used for many different, to produce different products, okay. So benzyl penicillin is the first antibiotic to have been manufactured in bulk. Hopefully you remember, excuse me, how penicillin was discovered, okay. So it can be prescribed for use in treatment, but it is also used as an input material for some semi-synthetic antibiotics. So remember, when we were looking at the benefits of microorganisms, one of those benefits was that we are able to generate raw material that can then be used in further production of, or they can be further modified to give us more beneficial material, okay? Because remember, the good thing, or even another advantage we talked about, was the fact that microorganisms are able to carry out stereospecific modifications and regiospecific modifications, okay? So that if you have a scenario whereby you want a molecule that is actually, whose stereochemistry is specific, then usually carrying out these modifications or synthesis can become very costly, okay? And one of the things that has been realized is microorganisms are actually able to do this quite easily, okay? So it's possible for you to generate a raw material and then modify it to actually a more effective compound, okay? So processes used in the production of benzyl penicillin are difficult to optimize, and no two apparent identical batches will ever be entirely the same. This is because living cell populations change both quantitatively and qualitatively throughout the production cycle. So again, remember, with the ideally what you would want is that you're able to generate the exact kind of material, okay? But unfortunately, in this case, you're not dealing with machines. You're not dealing with chemicals in the lab, okay? So that in this case, usually it is because these are microorganisms, okay? They are not exactly the same as with every previous batch or preceding batch, so that usually, even though you're producing the same material, you will actually have small differences here and there. But of course, the main thing you want is to get the parent compound, okay? If you're able to achieve that, that is acceptable enough, okay? So small changes in control parameters, such as fluctuation in air pressure or power dip, can potentially impact a batch, and the effect may vary dependent upon the age of the batch, okay? So for this reason, you want to be very specific, okay? You want a scenario whereby all the requirements are actually they remain constant, okay? You don't keep on changing the conditions. And the reason being, you want as much as possible to maintain the characteristics of your batch. There is also significant batch-to-batch variation in the complex nutrients commonly used in the fermentations. And no single product can exemplify all the important features of antibiotic manufacture. For example, benzyl penicillin being a beta-lactam, and the most industrial microorganisms used to make beta-lactams are fungi, okay? However, this is atypical of antibiotics as a whole, where bacteria, particularly Streptomyces species, are more commonly used. Again, of course, one of the important things that you need to be aware of is you need to, if you're going into this field, you need at least to be aware of the kind of microorganisms you'll be using, okay? You may use fungi, okay? Or you may use bacteria, okay? Again, depending on whether whichever you're using, again, here you can see that you may not get exactly the same thing, okay? These antibiotics can be made by total or partial chemical synthesis, where the starting material is generated through fermentation. We already mentioned that. Batched fermentation processes are used. For instance, processes where sterile media in a vessel is inoculated, the broth fermented for a defined period, usually hours or days, the tank emptied, and then the broth extracted by downstream processing to yield the antibiotic. During the fermentation, nutrients, antiform agents, and air are supplied. The pH is controlled and exhaust gases are removed. Okay, so again, you want to ensure that you do not get foam being produced in your tank, okay? Because if you get foam, remember these microorganisms are actually microscopic. You're not able to see them with your naked eye, okay? So it's possible for the microorganisms to get entrained within the foam, and then you will lose a whole batch of your microorganisms, okay? So you don't want foam in your production unit. After emptying, the tank is cleaned and prepared for a new batch, what is referred to as turned around. This is different from continuous fermentations, where sterile media is added to the fermentation with a balancing withdrawal of broth for product extraction. This can be advantageous provided the system can be run without contamination. One advantage is long fermentation runs for many weeks, hence greater productivity per vessel due to fewer turnarounds. Remember, when you consider turnarounds, what happens is the new batch is when you clean and then you prepare for a new batch, the new batch has to be to take some time for it to get to the ideal production point, okay? So you actually, there is some time factor there. But if you get continuous production, then it means that you're not interfering or interrupting your production, okay? Because all you're doing is making sure that you're adding media there to keep feeding your microorganisms, and their work is to keep producing, okay? In continuous culture, the growth rate can be held at an optimum value for product fermentation. It is therefore suitable for products whose synthesis is proportional to cell density, but is not generally an economical process for antibiotic production, where synthesis is not associated with growth, and there are additional concerns about strain degeneration. So again, it depends. Usually this will just depend on your microorganism. At what point are they most productive? Because remember, if you remember the graph in microbiology, okay, where you have the lag phase, okay, and then exponential growth, and then you get to the plateau phase. You need to be able to appreciate at what point do your microorganisms start producing the product that you want, okay? So you may have a scenario where they will indefinitely keep producing. In that case, you can have continuous production. But in the case where the microorganism have to be at a certain point to give you the best yield, in this case, that's what we are talking about, strain degeneration, okay? If you keep them for too long, then it may actually even affect production, okay? So looking at benzyl penicillin, the original microorganism that was used was Penicillium notatum, okay? It was isolated by Fleming. Hopefully you remember the Fleming incident where he discovered penicillin by chance, okay? The cultivation of this obligate aerobe was lengthy, labor-intensive, and prone to contamination. Now, remember, when we talk about, if you think of contamination of penicillins, hopefully one main thing comes to mind, okay? What comes to mind when you think about penicillin contamination? Relin, does anything come to mind? Okay. So anyway, usually with the penicillins, you don't want them to get contaminated because it's possible that the contamination they are getting could actually be beta lactamase producing microorganism, okay? So that if they get contaminated with a beta lactamase producer, what will happen is that your whole batch will actually be destroyed, okay? So that is usually one of the concerns, okay? A higher yielding organism, a microorganism, which was Penicillium chrysogenum, was isolated from an infected cantaloupe melon. This organism can be grown in deep fermentations in sealed tanks under stirred and aerated conditions in vessels as large as 250 meters cubed. Okay, so again, as you look at production, you want to get to a point whereby it's possible for you to produce a product with the least limits, okay, as much as possible. For instance, you can imagine being able to culture, okay, or cultivate the the initial notatum, okay, the cost would be very high, okay, especially if you need to actually give it the conditions, okay. But if you are to get a microorganism that does not is not very strict in the conditions that are required, then of course it becomes easier with time, okay. And hopefully you remember, I mean, most of this can actually be done through biotechnology, where you can even alter the microorganism to give you the right characteristics. So through selection procedures, manufacturers have evolved production strains. Large numbers of isolates have been screened to identify high can produce over 20 grams per liter of culture compared with the 2 milligrams per liter produced by the wild strain. So general requirements for inoculum preparation and handling. So again, remember, as we said, we don't want to have a scenario whereby we get contamination of our pharmaceutical product, okay? Because if you get contamination in this case, especially during production, you may get our biggest care for in this case is production of beta lactamase, okay? Because if you get that such organisms contaminating your product, what will happen is that you end up with no pharmaceutical product, okay? By now, all of us know that for your penicillin to actually work, it must be the lactam ring, the beta lactam ring must be intact, okay? But if it is not intact, then the penicillin will not actually be effective, okay? So should you get a microorganism that is able to break that ring, you can imagine producing a whole batch and then realizing that the whole batch actually has it has been degraded, okay? The beta lactam has been broken, okay? So it's important that you actually ensure sterility. And how is this done? All operations are carried out in laminar flow cabinets. Hopefully you remember what a laminar flow cabinet looks like, okay? This is to prevent introduction of contamination through air, okay? If you remember laminar flow cabinets, you have air flowing from one direction to another, okay? And some of these are even more advanced, such that you only have a place where your hands get in, okay? The whole cabinet is, your whole work is being done within the cabinet, okay? And you have a place where your hands actually are the ones that get in, so that you will not be able to breathe onto your product, okay? So hopefully you remember something like that. Let me see if I can share my screen and remind you what it looks like. So hopefully you can see the lady in the picture, okay? This is an example of a laminar flow cabinet, okay? So she's sitting in front of a cabinet, and you can see, I'm not sure you are able to see, but right where her hands are going in, it's basically just holes, okay? Her hands are going, she has inserted her hands into a cabinet. There's a place where the hands go in. She's gloved, okay? And so there's a glass in front of her. So she's not breathing onto whatever she's handling, okay? And assuming this was a hazardous material, if it is producing fumes, she's not inhaling the fumes, okay? So within a laminar airflow cabinet, what happens is air is flowing from one direction to another, and this air is sterile, okay? It can flow from right to left, left to right, up, down, okay? And all this is basically just to ensure that sterility is maintained, okay? So this occurs in rooms in which filtered air is maintained at a slight positive pressure relative to their outer environment. This prevents entry of contaminating microorganisms into the field of preparation. Operators will wear sterilized clothing and work aseptically. They have potential to introduce contamination due to hygiene and normal flora on the skin, which can get into the inoculum. So antibiotic fermentations are of strict necessity, pure culture aseptic processes without foreign growth. Contamination by other microorganisms is also avoided, okay? This could result in destruction of the whole product, like in the case of penicillin. So again, remember we said with penicillin, you want to avoid contamination with beta-lactamase producing microorganisms, okay? Because if you contaminate with those, you can be sure that you won't have any product, any useful product. Because, I mean, you will get the penicillin, but without the intact beta-lactam ring, it is useless, okay? So any additive added to the inoculum must be done aseptically to avoid introducing contamination. So how do we prepare the inoculum? Okay, we've looked at the prerequisites. So you need, everything must be done aseptically, everything must be done under sterile conditions. So during preparation, inoculum preparation is done to develop a pure inoculum in sufficient volume for production stage, for the production stage, okay? The inoculum should be in fast-growing logarithmic phase so that a high population density is quickly established. Remember, again, going back to that graph we talked about, you have the lag phase, okay? You have the exponential growth phase, then you have the plateau phase, okay? So you want to use the logarithmic or place where you have exponential growth, okay? So that you're able to actually achieve high population density. The final inoculum to the production stage is generally 1 to 10% of the total volume of the fermenter. If the fermenter is under-inoculated, there may be an extended lag before growth starts, and the fermentation period will be prolonged. This is uneconomical and may result in degenerative growth, which affects performance, quality, and cost. Again, of course, these conditions are not arbitrary, okay? One of the things that hopefully you know by now is that usually when these medicines are discovered, you optimize, okay? It's the way, like if you're working with a synthetic drug, remember with the when we were looking at building quality into your product, quality by design, one of the things that hopefully you remember is that during research, you optimize the conditions, okay? So you look at things in terms of what will happen if I increase temperature, for example, okay? What will happen if I increase pH, for example, okay? You may have increase in production up to a point. So that now when it comes to optimization, what you will do is you will have 2 points of reference where you'll say pH, for instance, must be above 4, but below 7, for example, because during your research, you are able to see that below 4, okay, there is basically no activity. And then maybe above 7, again, you have issues, okay? But within these limits, you're able to get optimal activity, okay? So again, it's the same thing here. The conditions that have been delineated are actually not arbitrary. These are conditions that have been found to be the most effective for the production of high volumes of penicillin, okay? So the inoculum stage media is designed to provide the organism with all the nutrients required for that stage. Adequate oxygen is provided in the form of sterile air, and the temperature is controlled at the desired level. Before they are transferred to the next stage in the progression, they must be free from foreign growth, and growth has to have occurred up to a predetermined cell density. Typical of fungi, the organism grows as branching filaments. By the time that the culture has progressed to the production stage, it is very viscous. Okay. So again, remember these are microscopic. Okay. So you may not be able to see with your naked eye, but basically you can see that the product is viscous. Okay. If you observe it under the microscope, you can see that it has a lot of branching filaments, but you may not be able to see this with your naked eye. So the fermenter is a closed vertical cylindrical stainless steel vessel with convexly dished ends, and usually the capacities will vary, okay, from 20 to 25 to up to 250 meter cubed capacity. Okay, so these are huge, uh, vessels. Its height is usually 2 to 3 times its diameter. Okay, so again, this is an example of a fermenter. These are huge drums. Okay, they will have different, they've basically been constructed to facilitate fermentation and production of the antibiotic. So again, in your own time, you can look at, if you're interested. Of course, I don't expect you to cram this, but out of interest, you can look at the different parts, okay? Of course, you can see from this, these are powered, okay? You have a place for adding antiforming agent, okay? You have a place for you to be able to sample, okay? And then again, how you're able to cool this vessel, okay? And then you have air filters, because remember, this is an aerobic process, okay? So you need filtered oxygen, and then agitator, all this, okay? You can look at that in your own time. So as we said, this is an aerobic process, meaning it needs to occur in the presence of oxygen. Okay, so how is this oxygen supplied? Okay, the penicillin fermentation needs large quantities of dissolved oxygen, which is supplied as filtered and sterilized air from a compressor. Oxygen is critical to aerobic processes, and its supply is a crucial aspect of the fermenter design. Okay, so like from the design we have seen, there's they've allowed for a place where you can actually add air into the vessel. Okay, as oxygen is poorly soluble in water, steps are taken to assist its passage into the liquid phase and from aqueous solution into the cell, the latter being a particularly problematic process in viscous broth. One option is to introduce air into the bottom of the vessel via a ring spurger with multiple small holes rather than through a single large orifice. This breaks the airflow into smaller bubbles, which have a greater surface area to volume ratio and hence greater oxygen transfer. These bubbles lose oxygen as they rise up the tank, and at the same time, carbon dioxide diffuses into them. The vessel is kept under positive head pressure, which promotes dissolution of oxygen and in addition reduces chances of contamination. Then, of course, we have temperature control. This production of benzyl penicillin is sensitive to temperature, and a lot of metabolic heat is generated, and the fermentation temperature has to be reduced by controlled cooling. So again, from the diagram with the vessel, you could see that a mechanism has been put in place to ensure that the vessel can be cooled. Okay, so this heat transfer is achieved by circulating chilled water through banks of pipes inside the vessel. These coils consist of continuous lengths of pipe welded in a shallow spiral around the vessel. The cooling water system is also used to cool batched media, which has been sterilized in the vessel prior to its inoculation. Then we have deforming agents or anti-forming agents. Okay, microbial cultures may form when they are subjected to vigorous mechanical stirring and aeration. If this forming is not controlled, culture is lost by entrainment in the exhaust gases. So again, consider that these microorganisms are microscopic, meaning you will not be able to see them with your naked eye. They are very tiny. Now, consider a soap bubble, okay? If you have a soap bubble, it's possible for that soap bubble to move away and carry a lot of microorganisms with it. Because remember, the microorganisms are very small. You're not able to see them with your naked eye. Okay. So now, in this case, if you're actually uh, getting a lot of foam being formed in your vessel, it's possible for many of these microorganisms to attach or get what we are referring to as entrainment in the exhaust gases, okay? Because they get attached to this foam. And then now, as you're releasing your carbon dioxide out of the vessel, you lose part of the culture, okay? So for this reason, you have to add this antifoaming agents, okay? And they basically reduce the formation of foam. So automatic systems are installed for detecting incipient foaming. These automatic systems also temporarily apply back pressure to contain the culture within the vessel. They are also applied in the aseptic addition of deforming agents or antifoaming agents. So instrumentation is fitted to provide a continuous display of important variables such as temperature and pH, the power used by the electric motor, airflow, dissolved oxygen, and exhaust gas analysis. So what we are saying is that these vessels come equipped with, you can see that it has many parts to it, okay? And usually they will have a panel that is able to show you what is happening, okay? A place for the power, it will have show a place showing you the pressure, okay? It will have a place showing you the temperature, okay? And all that. That's what we are basically saying when we talk about instrumentation, okay? Manual or computer feedback control can be based either directly on the signals provided by the probes and sensors or on derived data calculated from those signals, such as the respiratory coefficient or the rate of change of pH. Analysis of exhaust gases can provide valuable physiological information. So again, as we said, most of this is built into this vessels, okay? You're able, because of the built-in sensors and the continuous display of important variables, it's an easy system to monitor, okay? Then we have media additions, okay? Okay, as you're carrying out your fermentation processes, one of the things you'll realize, remember initially we talked about microorganisms having different requirements for nutrients. Okay, so again, one of the things you will notice is for some of these microorganisms, what has been realized is that if you want to promote production of a specific product, you can feed the microorganism with a raw material that they require, and then they will give you the product that you want. Okay, so not all the nutrients required during fermentation are initially provided in the culture medium. Some are sterilized separately by batch or continuous sterilization, then added while the fermentation is in progress. These nutrient additions usually occur via automatic systems that allow a preset program of continuous or discrete aseptic additions. This type of fermentation is called fed batch. Again, of course, one of the beauties of this, the fact that this can be automated, is you reduce contact with humans, okay? Because remember, uh, you're able to appreciate that we walk around with normal flora, okay? And we don't want to contaminate these batches. So the fewer the human contact, the better for this batch, okay? So that's why you will find most of this equipment are actually automated, okay? Everything can be done automatically by just feeding the required information. Then we have transfer and sampling systems. Uh, excuse me. We were looking at transfer and sampling systems. Aseptic systems are used in the following steps. So again, as we said, whatever you do in this whole process, you have to make sure that it is done aseptically, okay? Whether you're carrying out sampling or you're transferring, because if you look at the vessel, like in this case, you can see the vessel actually even has a um, sampling line, okay? So that it's possible for you to access the contents without going in, okay? So that as we said, the main aim here is actually to reduce contact, because every time you come into contact with this product, you're increasing the chances of contamination, okay? So all these steps have to be done aseptically, okay? To transfer the inoculum into the vessel should be done aseptically to allow removal of routine samples during fermentation, for early harvesting of aliquots when the vessel becomes full as a consequence of media additions, to transfer the final contents to the extraction plant when fermentation is complete. So all these have to be done aseptically, okay? Remember what we said. You don't want contaminants getting into your fermentation vessel. Asepsis is assured by engineering design and by steam sterilization, which must reach all parts of the vessel and associated pipework. Any pockets of air or rough surfaces that steam does not penetrate could act as reservoirs for foreign growth. Sampling is essential to monitor the amount of growth, the running levels of key nutrients, and the penicillin concentration. It is necessary also to check that there has been no contamination by unwanted microorganisms. Because remember, you may get to a point, if you find contamination, in some cases, you may actually destroy the whole batch, okay? It will no longer be acceptable. So these are things that you have to take note of. So if growth in the fermenter proceeds unchecked, okay, at the rate prevailing in the seed stages or the initial stages, the culture would become very dense and the available aeration would no longer be sufficient to maintain penicillin production. So again, remember, we said that this process is aerobic, okay? It needs oxygen for the process to happen as it should. Now, one of the things that you need to do is to control the growth of this, the fungi, okay? Because if you don't control it and there is uncontrolled growth, usually what will happen is you will have some places where you have anaerobic growth, okay, where oxygen is not reaching, okay. So usually you have to control growth so that it's not very dense. Should oxygen availability fall below a critical level, benzyl penicillin biosynthesis is greatly reduced, although culture growth continues. So you can see the little things that we talked about. And most of these, remember we said they are not arbitrary. These are conditions that were actually, they came up with them during research, okay? And as we said, usually you want to see what is the effect of this parameter, what is the effect of this one. So like in this case, what was realized is that oxygen is key, okay? If you reduce the oxygen, what will happen is this fungi will continue to grow. Unfortunately, it will continue to grow, but it will not produce the required amounts of benzyl penicillin. Okay. So accordingly, conditions are so adjusted that fast growth is achieved only until the cell population has reached maximum density that the vessel can support. Further net growth is constrained by deliberately limiting the supply of a key nutrient, usually a sugar. The cells can then be stimulated to an overproduction of benzyl penicillin while restricting the amount of growth, and a stable, highly productive cell population can be sustained. So again, if you look at all these are results of research, okay? What is the effect of oxygen on the on this production cycle, okay? How does it affect growth of the fungi, okay? And how does it affect production of benzyl penicillin? Okay, so you have all those effects. Okay, and then so at this point, of course, they realize that if they limit the supply of sugar, you limit the growth of the fungi. Okay, and then now you can stimulate production of benzyl penicillin. Okay. The medium initially is initially batched into the fermenter. Uh, this medium usually is a complex one, okay, but designed only to support the desired amount of early growth, okay. So again, remember, we said you want growth to occur up to a point, okay? But beyond a certain point, if you have too much growth, you may actually, it limits production of benzyl penicillin, okay? So initially, whatever is put into the fermenter, you want growth up to a certain point, okay? The principal nitrogen source is corn steep liquor, a byproduct of the maize starch producing industry. This material is recognized as valuable in many fungal antibiotic media. The primary purpose of corn steep liquor is to supply cheap and readily available nitrogen. This CSL or corn stiplica also contains a useful range of carbon compounds such as acids and sugars, together with inorganic ions and growth factors. Due to this, it is virtually a complete growth medium in itself. So the constiplica is a complex nutrient. It is not chemically defined. It is derived from natural products and with significant batch-to-batch variation. It is therefore one of the reasons why no two fermentations are ever absolutely identical. Again, remember we said your ideal would be that you get identical fermentations. But remember, these are living microorganisms, okay? So whatever you give them as feed will determine the kind of fermentation, okay, that you will get. So again, simply because this CSL is a natural, it's a complex nutrient, means that you will not be able to actually get identical fermentations, okay? In recent years, a number of manufacturers have moved from using this relatively cheap but chemically undefined complex nutrient to defined media, thus reducing variability in their fermentation processes. The batched medium contains subsidiary nitrogen sources and additional essential nutrients such as calcium added in the form of chalk to counter the natural acidity of the CSL. Magnesium is also added, sulfate. So all these are added as source of nutrients, okay, for the fungi, okay? The medium is sterilized with steam at above 120 degrees Celsius, either in the fermenter itself or in ancillary plant, which may be worked continuously. The sterile media, and so the previous ones we were talking about were the batched media, okay? What is fed at the beginning, okay? Then we have fed nutrients, okay? Remember we said not everything is actually put in the fermenter at the beginning. Sometimes you may have to add nutrients as time goes, okay? So you want to look at these fed nutrients. The sterile media is stirred and aerated, and its pH and temperature are adjusted to the appropriate values. It is then inoculated and the growth phase begins. The initial carbon source is sufficient in quantity to maintain early growth, but not sufficient to provide the energy that penicillin production and maintenance of the cell population need during the rest of the fermentation. Carbon for these subsequent stages is fed continuously in such a way as to limit net growth. Remember we said you don't want to you don't want to overfeed, because if you overfeed them, then you will have overgrowth, and then with overgrowth you don't get uh, it limits production of benzyl penicillin, okay? Sucrose or glucose is used, possibly as cheaper impure forms, such as molasses or starch hydrolysate. Okay, so remember, in this case, you're feeding them molasses or starch hydrolysate, but what the microorganisms are drawing from them is sucrose and glucose. Okay, as the concentration of residual sugar in the broth is too low to measure, the rate of feeding has to be learned by experience and modified on the basis of systematic observation. But again, remember, we said with these automated vessels, it's possible to actually control this feeding, okay? An alternative way of attaining carbon limitation without the complication of a carefully monitored carbon feed rate is to supply all the carbohydrate at the outset as lactose. The rate-limiting hydrolysis of lactose to hexose is then relied upon to give a steady, slow feed of assimilable carbohydrate. Okay. Originally, all benzyl penicillin was manufactured using lactose in this way, and some manufacturers still prefer these techniques. We talked about these elements: calcium, magnesium, phosphate, and trace metals added initially. These are usually sufficient to last throughout fermentation. The microorganisms, however, need further supplies of nitrogen and sulfur to balance the carbon feed. Fed nitrogen is often supplied as ammonia gas, and the whole system is a balanced one. The carbon and nitrogen feeds will do two things: satisfy the organism's requirement for these elements in the correct molar ratio, and maintain an adequate reserve of ammonium ion and contribute to pH control. The carbon metabolism being acidogenic and balanced by the alkalinity of the ammonia. Sulfate is usually supplied in combination with the sugar feed, and by obtaining the correct ratio, there is a balanced presentation of sulfate with an adequate pool of intermediates. So again, I don't expect you to cram this whole process, but I do expect you to appreciate, the role of these steps, or the roles of some of these nutrients, okay? Like when we come to addition of phenyl acetic acid. Why are we adding phenyl acetic acid? And what would happen if we did not add phenyl acetic acid? Okay, that is what I want you to be able to appreciate. Because remember, the main objective of this part of the unit is for you to appreciate that microorganisms have actually been used in the production of pharmaceuticals. But the process may uh, the processes may be similar in a way, but for each of those pharmaceutical products, there are differences. Okay, so it's important for you to be able to appreciate, like in this case, as we're looking at benzyl penicillin, okay, you're able to appreciate that for some microorganisms, you carry out batched processes. For others, you carry out continuous processes, okay? And why is that, okay? Like for the benzyl penicillin, we have said you don't want continuous production because microorganisms will get to a point and start degrading or going down, okay? So that production will be reduced. So the main aim here is not uh, to cram, okay, but to appreciate every step that we are talking about. So the growth phase passes rapidly into the antibiotic production phase. The optimum pH and temperature for growth are different from those of penicillin production, and therefore there are changes in the control of these parameters. Addition of phenyl acetic acid by continuous feed marks the onset of the production phase. All feeds are sterilized before they are metered into the fermenter. So again, remember we said everything has to, everything is, most of these things are automated, okay? You are trying to achieve optimum pH, optimum temperature, okay? And you're able to tell at what point, because remember we said growth occurs up to a certain point and then you limit growth. How do you limit growth? By limiting sugars, okay? And then now, because if you don't limit the sugars, growth will happen indefinitely and you will have a limit to the amount of oxygen that is able to penetrate into your fungi. Okay, so you limit all that, and then now when you get to the point when you have achieved optimum growth, you now feed phenylacetic acid, which marks the production phase. Okay, and as we said, everything that goes into the fermenter must be sterile. Contaminants resistant to antibiotic are detrimental should they find their way into the fermenter, and their effects are so damaging that prevention is of paramount importance. Any foreign growth has the potential to disrupt fermentation processes with both quality and financial implications. For example, a resistant beta-lactamase producing fast-growing bacterial contaminant can destroy the penicillin already made, as well as consuming nutrients intended for the fungus, causing loss of pH control and interfering with the subsequent extraction process. Okay, so you can imagine if you get another contaminant coming in producing unwanted substances. Okay, it means that when you go to isolate your product, you will actually get contamination even in your product. Okay, of course, we've talked about the scenario whereby you break the beta lactam ring and then now your product is useless. Okay, it will not be effective. Therefore, the key requirement in both design and operation of all stages of the fermentation is that aseptic operations be maintained with growth of the process microorganism, but no unwanted foreign growth by contaminating microorganisms. So we've talked about phenylacetic acid marking the production of or the production stage. Okay, phenylacetic acid supplies the side chain of benzyl penicillin. Without the phenylacetic acid, the microorganisms synthesize only small quantities of this penicillin. So the phenylacetic acid stimulates benzyl penicillin biosynthesis, and it also suppresses the formation of other unwanted penicillins. Phenylacetic acid is, however, expensive, and high levels of it are toxic. So again, remember we said most of this information is gathered during research. Okay, you're looking at a scenario whereby you realize that the microorganisms require, uh, at some point, they make phenylacetic acid, okay? And it's the rate limiting step. So the question is, what happens if you add them more phenylacetic acid? You increase quantities of benzyl penicillin produced. Do we add this molecule indefinitely? Okay, do we keep adding it uh, in increasing concentrations? No, because what was realized is that at a certain point, okay, the amount exceeding a certain amount becomes toxic to the microorganisms. Okay, so again, these are things that are discovered during research. Okay. Therefore, the feed is controlled to supply an adequate standing level of phenylacetic acid without approaching the toxic limit. The feed is reduced just before the end of the process so that the amount of unused and usually irrecoverable precursor in the final culture is not excessive. Okay. So how do you terminate production? Okay, because remember we said this is a batched process. Okay, you terminate production, you recover the product and start again. Okay, so the fermentation process has to be stopped. Several factors have to be taken into account to terminate the process. Often, a manufacturer will find it appropriate to harvest shortly after the first signs of a faltering in the efficiency of conversion of the most costly raw material into antibiotic. Then we have extraction or removal of cells. At harvest, the benzyl penicillin is in solution extracellularly together with a range of other metabolites and medium constituents. The first step in downstream processing is to separate the cells from the liquid broth by techniques such as filtration, ultrafiltration, or centrifugation. This stage is carried out under conditions that avoid contamination with beta-lactamase producing microorganisms, which could lead to serious or total loss of product. Then we have extraction, which involves isolation of benzyl penicillin. This benzyl penicillin is isolated through solvent extraction, which is generally accepted, although other methods are available, including ion exchange chromatography and precipitation. In aqueous solution at pH of 2 to 2.5, there is a high partition coefficient in favor of certain organic solvents such as amyl acetate, butyl acetate, and methyl isobutyl ketone. Okay. The extraction has to be carried out quickly as benzyl penicillin is very unstable at these low pH values. The penicillin is then extracted back into an aqueous buffer at pH 7.5, the partition coefficient now being strongly in favor of the aqueous phase. The solvent is recovered by distillation for reuse. Benzyl penicillin is then crystallized, blended, and dried. The downstream processing stages are designed not only to recover the antibiotic, but also to ensure that it is of the appropriate potency, purity, and that this is achieved by cost-effective processes. Okay, so again, at this point, these are basically lab processes. Okay, you have gotten the benzyl penicillin, you want to get it in pure form, but remember, it comes together with other components. Okay, it will come together with metabolites, medium constituents, okay, and all that. So you want to be able to isolate it so that you get it in pure form. Then it undergoes further processing, which involves formation of an appropriate salt, usually followed by treatment to remove pyrogens and sterilization. So again, we've talked about pyrogens time and again. Hopefully by now that has stuck. Okay. The salts can be used as an input to semisynthetic beta-lactam antibiotic manufacture or for clinical use in its own right. Okay. Generally, sterilization is achieved by filtration through pure metals, or rather, generally sterilization is achieved by filtration, although pure metal salts of benzyl penicillin can be safely sterilized by dry heat if desired. Again, remember we talked about, you guys looked at pre-formulation, okay? And one of the things you carry out in pre-formulation, pre-formulation basically involves getting to know the physicochemical characteristics of your product, okay? So like for benzyl penicillin, what you would want to know is after production, is it stable to heat, okay? And here you want to know whether it can be sterilized and what methods are available that can actually be used for sterilization. So like in this case, there are two methods. You can use dry heat or you can use filtration. For parenteral use, the antibiotic is packaged in sterile vials as freeze-dried powder to be reconstituted before use or as a suspension. For oral use, it is prepared in any of the standard presentations such as film-coated tablets. Suching tests are carried out on a significant number of random samples of the finished product to ensure that it satisfies the stringent requirements. Okay, so again, like any pharmaceutical product, these are standard tests that will be carried out. And again, as we said, all this is reached upon during pre-formulation. Okay, the finished product is taken through quality control tests for quality, And all this is actually tested to check for potency, purity, okay? As we said, freedom from pyrogens and sterility. Then we have, remember we said benzyl penicillin can actually be used as it is, or it can be uh, changed, okay, used as a raw material, okay? So let's look at production of penicillin V, okay? How do you uh, produce penicillin V instead of benzyl penicillin, okay? So like in this case, you actually are able to produce penicillin V by, instead of, remember we said at some point we said when your inoculum or your growth has reached the required amount, you limit growth by reducing the uh, carbohydrate, and then you um, you add phenylacetic acid, and that is to signal production of benzyl penicillin. Okay, but if you wanted to produce penicillin V, now you will not feed the growth with the uh, phenylacetic acid. Okay, but you will instead feed it with something else. Okay, so by the addition of different acyl donors to the medium, different penicillins can be biologically synthesized. Okay. For example, penicillin V is made by a similar process to benzyl penicillin, but with phenoxyacetic acid as the precursor instead of the PAA that we saw earlier. The microorganism used is still the penicillin chrysogenum, okay? But at the point where you add the PAA, in this case, you add phenoxyacetic acid, okay? A manufacturer may use the same mutant strain to make both products or may have different mutants for the two penicillins. Parallel situations of a single organism producing more than one natural product occur with other types of antibiotics as well. For example, strains of Streptomyces aurefaciens are used for both chlor-tetracycline and dimethyl chlor-tetracycline fermentations. So again, remember we said we are just looking at this as an example, okay, so that it can give you an idea of generally what would occur in a fermenter, okay? Like benzyl penicillin, penicillin V is still used in its own right, but can also be used as a starting material for the manufacture of semi-synthetic penicillins, which cannot be made by direct fermentation. Then we have production of cephalosporin C. It's possible to convert penicillin V or benzyl penicillin to a cephalosporin by chemical ring expansion. The first generation cephalosporin, which is cephalexin, for example, can be made in this way. Most cephalosporins used in clinical practice, however, are semi-synthetic, produced from the fermentation product cephalosporin C. Okay, so now from this point, remember we said at this point you have penicillin V or benzyl penicillin, and okay, now you're able to get cephalosporin C. Okay, and here you can actually uh, you can actually, as we said, either use the benzyl penicillin as is or penicillin V as is, or you can take them through synthesis from this point on, okay, where now you get semi-synthetic products that can be used for treatment. Any question? So next, I want us to look at bacterial resistance to antibiotics. So you guys are aware that as much as we human beings are trying to destroy bacteria and infective microorganisms, they also are trying to survive, okay? So as we come up with the chemicals to destroy them, they also, with the intention of surviving, they keep coming up with ways of circumventing, okay, or avoiding the means that we are using to actually kill them, okay? And because of that, you get resistance, okay? So what is bacterial resistance, okay? We will look at this, and then we will look at different antibiotics and the kinds of resistant processes they will actually go through and some of the effects, okay? So antibiotic resistance has been around for as long as antibiotics have been used to treat infection. So remember, initially, I mean, from the first time it was discovered that these chemicals can actually kill microorganisms, even the microorganisms with the aim of surviving started coming up with ways of ensuring that they are not killed, okay? That is the origin, okay? So antibiotic resistance reflects the attack and counterattack of complex microbial flora in order to establish ecological niche and survive. Early treatment failures with antibiotics did not represent a significant clinical problem because other classes of agents with different cellular targets were available. So what we are saying is when you have a huge class of available antibiotics, okay, such that you're not, I mean, you have different classes to choose from, it's not a big deal if you get resistance from one compound, okay? But if you get to a scenario whereby now you're getting resistance to different microbes, or you're getting resistance to different antibiotics at the same time, okay, then it becomes a matter, okay? The emergence of multiple resistance antibiotic agents is the major cause of concern in antibiotic use today. Due to the introduction of extended spectrum agents, there has been a huge selective pressure in favor of multiply resistant species. There has also been a sharp decline in the introduction of agents acting on new cellular targets over the last 30 years. So again, one of the things that you'll appreciate is that when you get emergence of resistance to several antibiotics. Okay, what that means, it's possible to get to a scenario whereby you have a patient, they have an infection, and you try so many agents, and all of them seem to be resistant. Okay, which can be quite scary. Okay, there have been cases whereby patients have died because nothing that was available was actually helping them. Okay, the infection they had was not responding to any of the uh, antimicrobials available. Okay, so that becomes a matter of concern. Initially, when there were many uh, classes still effective, it wasn't an issue. But now, when we keep getting multiple drug resistance, it becomes a matter of control. Okay, like I believe I've mentioned this in this class. Like one time we had a scenario whereby we had a baby who was had an infection, and for some reason, when we looked at the resistant pattern, we realized that infection was actually resistant to most of the antibiotics that we had available. Okay, and one of the things that we found that was effective, or the microorganism was sensitive to, was actually fluoroquinolone. Okay, what is your concern when you think about fluoroquinolone and children? What comes to mind? Uh, Bruce,
Hello.
hello.
I believe fluoroquinolones, when given to children, has side effect on the development of their cartilages, so it should be avoided in children.
Yes. So theoretically, that's correct. You're told that you do not give fluoroquinolones to children because it will actually affect development of cartilage. But now, what would you do in this case, Rebecca?
Hello?
Hello.
Because it's the only drug that tends to have effect on the baby, that is what you could give, because we again have to save life. Then know what next. Yeah.
Yes, and actually, that's exactly what we did. Okay, we gave the baby ciprofloxacin, and the infection was resolved. So, what am I saying? Sometimes, as you get into practice, you realize that sometimes what you're taught in school is basically just that theory. Okay, it may not always apply to what is in practice, because remember, as we said initially, you're given the power to read and do all that pertains to your degree. Okay, so hopefully as you go out there, you're able to make independent decisions, because at this point we have a baby, whatever infection they have is resistant to everything that we have available. Okay, then we have this drug, we have tested, we have seen that there is sensitivity. What do we do? Okay, first we need to think about saving life, and then after that we can consider. Okay, but again, if you look at most of this cartilage development in some of these cases, especially for young people, usually you'll be told limit movement. Okay, and just because at this point, okay, you have no other option. Okay, so again, that is something that you may encounter when you get into practice. The other thing, of course, it's not part of this class, but one of the things you'll realize is you will not get this fluoroquinolones in pediatric dosage. Okay, you will not find a syrup of a fluoroquinolone. So again, it brings you to your, uh, you know, lectures in class. Okay, how do you handle such a situation, because the manufacturer does not recommend use of this drug in pediatrics. Therefore, there is no pediatric formulation. You have this baby who is supposed to take this drug. How do you handle it? Okay. So again, those are just things that you have to keep in mind. Remember, we said we give you theory, okay, but you may get to the ground and realize that you need a little more than just theory to actually do all that appertains to your degree. Okay, so anyway, let's look at bacterial resistance. Notable resistance has been seen in the following microorganisms. We have gram-positive microorganisms such as methicillin-resistant Staph aureus, commonly referred to as MRSA. We have coagulase-negative Staphylococcus. We have glycopeptide intermediate sensitivity Staph aureus. We have vancomycin resistant Enterococcus, okay, penicillin resistant Streptococcus pneumoniae. So concerns among the gram-negative organisms include multi-drug resistance, Pseudomonas aeruginosa, Trophomonas maltophilia, and Acinetobacter baumannii. And then we also have members of the Enterobacteriosis with extended spectrum beta-lactamases. Okay. Multi-drug resistance in the acid-fast bacilli, Mycobacterium tuberculosis and Mycobacterium avium complex, pose major health threats worldwide. Okay, so one of the things that you'll realize is that with the resistance, there is danger of losing your patient. Okay, it can be very scary when you are in a hospital. A patient comes, you can see this is an infection, but whichever antibiotic you give them, the patient is not responding to. Okay, this can be quite scary because you can clearly see the patient ebbing out, and there's nothing you can actually do about it. Okay, and yet this is not Stone Age era. This is actually an era where we actually have antibiotics. Okay, just that the available antibiotics are actually not in that scenario, they are actually not effective. Okay, so what are some of the origins of resistance? Remember we said it was a big thing when antibiotics were discovered, okay? Because it meant it was the end of people dying from infection, okay? But as we said, our opening statement, immediately antibiotics were discovered, microorganisms started coming up with ways of acquiring resistance. Okay, so what are some of these origins of resistance? First, we have innate resistance. This typically reflects variations in the structure of the cell envelope. Remember, if you look at microorganisms, some of them the structure of the microorganism, you have the cell envelope, and this is basically from your biology. The cell envelope is supposed to limit the entry and exit of substances in and out of the cell. Okay, it's supposed to enclose the cell organelles and limit entry of water and other substances. Okay, now when we'll be looking at mechanisms of resistance, or rather mechanisms of action, one of the things you'll appreciate is that some of these antibiotics act on the cell envelope. Okay, they compromise the cell envelope, and by so doing, they will compromise the function of that cell envelope, which means that the microorganisms will die because now it is not able to prevent entry and exit of substances from the cell, and now that will compromise the cell. Okay, so this microorganism alter the cell envelope such that now the antibiotic is no longer able to interfere with that function. Okay. Then we have phenotypic resistance, which resistance or reduced susceptibility may be phenotypic such that we get adaptation to growth within a specific environment. So in this case, you don't have genetic modification. Okay, the microorganism has not genetically modified. It's just that because of its presence in a specific area within the body, it adapts itself such that it is not the antibiotic is not able to affect it. Okay, but if you isolate that microorganism, okay, and allow it to grow in the lab, you find that it is still susceptible. It's just that within the body there is a way it is able to evade the antibiotic. Okay, a characteristic of such phenotypic resistance is reversion to antibiotic susceptibility upon subculture in conventional laboratory media and failure to isolate genotypic resistant mutants. So with this kind of resistance, if you isolate this, if you grow these microorganisms, okay, in the lab, and you test them against this drug, you find that they are actually susceptible to the drug. It's just that in the body, they are able to kind of hide from the drug, okay? But when you go to the lab and try to get whether they have any resistant mutants, you find that they don't. And in fact, in the lab, they actually are susceptible to the antibiotic. Then we have another origin of resistance is acquisition of genetic material. And this involves acquisition of conjugative plasmids containing resistance genes. The origins of antibiotic resistance genes are unclear. So resistance can be achieved by horizontal acquisition of resistance genes mobilized via insertion sequences, transposons, and conjugative plasmids by recombination of foreign DNA into the chromosome. So again, in this case, this bacteria or microorganisms that were sensitive to antibiotic will basically acquire genes, okay, from the environment. And when they acquire these genes, they basically acquire resistance, okay? Then we have mutations in different chromosomal loci, which can cause resistance. Many resistance genes will have been derived from the diverse gene pool, or rather, which have been derived from the diverse gene pool present in environmental microorganisms, most likely produced as protective mechanisms by antibiotic-producing organisms. So again, these microorganisms will undergo mutations, okay? And when these mutations happen, they cause resistance in these microorganisms, okay? Genetic exchange is likely to arise in soil and the general environment as well as the gut of humans and animals. Rapid mutation can occur, and there's clearly a heavy selective pressure resulting from overuse of antibiotics in medical practice. So remember again why we keep on saying there's no need to use antibiotics unnecessarily, because you increase this selective pressure, okay? The body has a natural mechanism of protecting itself. We have normal flora which prevent overgrowth of harmful microorganisms. But if you keep taking antibiotics indiscriminately, usually what happens is you may end up killing the normal flora, which means that now the harmful microorganism no longer have any other organism to compete with for nutrients and space, so they now are able resistance. I believe this last slide for today. So genetic, the soil and general environment, as well as the gut of humans and animals. Rapid can occur, and there is clearly a heavy from overuse of antibiotic in medical practice, as we mentioned. Agricultural and vet use of antibiotics also makes an important and unhelpful contribution. Remember, one of the areas that was not being looked at so much was the use of antibiotics in vet products, okay? And remember, I told when you're treating your animal, get rid of, for example, or get rid of the eggs. But in most cases, farmers don't do that. So you guys know that some of these antibiotics will be released into the milk, will be released into eggs, okay? And if human beings feed on this, what happens is you're getting small amounts of antibiotics into your system, and this can actually result in development of resistance, because now when you keep feeding these microorganisms small amounts of antibiotics that are not going to kill them, but are giving them a chance to actually develop resistance, okay? So that has been an issue. The mutation process is not a static event, and a complex network of factors influences the rate and type of mutants that can be selected under antibiotic selective pressure. Antibiotic concentration, physiological conditions such as nutrient availability and stress can each regulate mutant or mutation rates. Not every mutation in a gene that encodes an antibiotic target leads to resistance. Remember, sometimes even resistance can cause the microorganism to be even more susceptible to a drug, okay? But the resistance, the changes that we are concerned with are changes that would cause the microorganism to become resistant to our drug. Resistance only occurs by mutations which are both permissive, that is, not lethal, or leading to an unacceptable reduction in fitness or ability to cause infection, and able to produce a resistant phenotype. So it's possible for a microorganism to undergo mutation, but the mutation is not compatible with life. So the mutation kills it, okay? Or it might cause reduction in fitness, okay? That is a mutation that is not fit for the animal, for the microorganism, okay? But there are mutations that can actually make the microorganism more stronger, so that now it is able to resist any clinical intervention that you may give. Those are the resistance. That is the resistance that we do not want, okay? Any question? So if there are no questions, we will stop there for today. Then next time we will look at the mechanisms of resistance, okay? And as we look at mechanisms of where do they occur, okay? Because you may find that for one, say like for tetracycline, they are affected by efflux, okay? Or like bind onto penicillin binding protein. But when it gets into the microorganism, it finds that the binding protein is altered, so it will go to bind, okay? So we'll start that and see how these different mechanisms affect the different antimicrobials, okay? So if there are no questions, we'll stop there for today. I'll see you next week. So I'm told I was speaking to Dr. Lily, and because I had been advised about some makeup, so probably your class rep can communicate when we can do these makeups, because I initially we had agreed on tomorrow, I believe, and Friday, but you can communicate and let me know